Have you ever tried to imagine finding one specific person in a city of millions? Now, what if that entire city was the size of a single drop of water? Well, that's the incredible, almost impossible challenge scientists face every single day, trying to isolate one specific cell. Today, we're going to look at a piece of technology that's an absolute game changer for doing just that.
Seriously, just think about that for a minute. If you're a scientist looking for a cure for cancer, how in the world do you find that one rogue cell that holds the key when it's just swimming in a sea of millions of perfectly healthy ones? It's a task that's, well, it's truly monumental.
You know, the classic analogy is a needle in a haystack, and honestly, it's the perfect description. For decades, this has been one of the biggest roadblocks in biology and medicine, you name it. Finding that one special cell is everything, but the haystack is just unbelievably enormous.
So yeah, let's start right there, by really digging into this fundamental challenge of isolating cells.
But what if you had a tool, kind of like a microscopic magnet, that could just go in and pull that exact needle out of the haystack for you? Well, that's exactly where Cell Sorter comes in. It's this amazing technology that uses high-powered imaging to find and then physically manipulate single cells, either by what they look like, their morphology, or by making them literally glow.
So let's break down exactly how this tech turns what seems like an impossible task into a totally routine procedure.
You know, the process itself is just remarkably elegant. It's all broken down into 4 simple, automated steps. First, the system scans the entire cell culture to create this huge, detailed map. Then, some really powerful AI software analyzes that map to pinpoint the exact cells you're looking for. From there, a tiny robotic micropipette goes in and precisely picks up the chosen cell. And finally, it deposits that single cell into a tube, perfectly isolated and ready for whatever analysis comes next.
And this really gets to the heart of it. It's not just about sorting cells in a general sense. It's about picking the exact cells you need. That incredible level of precision is what's opening up whole new worlds of research.
Okay, so how does it actually pull this off? What's going on under the hood? Let's dive into the key features that make the Cell Sorter technology so powerful and, frankly, so unique compared to the older ways of doing things.
What makes it so effective is this amazing blend of being super gentle but also incredibly versatile. The whole process is so delicate that the cells actually stay alive and healthy, which is absolutely critical for research. It can sort cells right out of a petri dish in just a few minutes, and it doesn't matter if they're floating around or stuck to the surface. Best of all, the whole system is modular, so it can actually grow and adapt as a lab's needs change.
Millions. Get this. In just 30 minutes, the system can scan millions of live cells, all while using a high-definition autofocus. Just try to wrap your head around the scale of that. It's like being able to read the cover of every single book in a massive library in half an hour just to find the one you need. It's incredible.
Now, to really appreciate the leap forward here, let's compare it to the old standard, a method called flow cytometry, or FACS, where FACS is really harsh on sensitive cells and has a tough time finding the super rare ones. Cell Sorter just shines. Because it uses high-res imaging to make its selection, it's way more precise and way more gentle. Plus, it's more compact and cheaper to run. I mean, it's a clear win in pretty much every category.
Okay, the tech is impressive, for sure. But what does it actually mean for the real world? This is where it gets truly, truly exciting.
First up, and this is a huge one, cancer detection. Being able to find that one rogue cell we talked about, that's been the dream for oncologists for decades, and this technology is making it a reality.
This allows for something that's called a liquid biopsy. So instead of an invasive surgical biopsy, doctors can use a simple blood sample. The Cell Sorter is so precise it can find those few circulating tumor cells, or CTCs, that have broken off from a tumor and are just floating in the bloodstream. By analyzing these cells, doctors can figure out if a treatment is working in real time. This isn't just an improvement, this is the future of personalized medicine.
All right, next up, let's look at maybe one of the most complex things in the entire known universe, the human brain. To even begin to understand it, we have to understand its individual building blocks, the neurons. So this tech enables something called single-cell transcriptomics. Now, what that basically means is it's like intercepting the real-time messages, the RNA, that are being sent out inside a single cell. By isolating one neuron at a time, scientists can listen in on these messages to figure out exactly what each cell is doing. This could unlock secrets about everything from how we form memories to diseases like Alzheimer's.
Of course, when you have this level of precision, developing new drugs is another major application. I mean, finding the right molecule to become a new drug is its own kind of needle in a haystack problem, right?
Here's how it works. Scientists can create millions of different versions of a protein, and then use the cell sorter to automatically find the single one that performs the best. This is how they develop these incredible tools called dreads. You can think of them like custom-built locks that get installed on a cell, and only a specific designer key, or drug, can open them. It gives researchers this unbelievable level of control, and it just massively speeds up the whole drug discovery process. Our immune system, it's like our body's own microscopic army, and understanding how those little soldier cells actually work is fundamental to fighting off infections and diseases.
With Cell Sorter, researchers can pull out a single, living immune cell and just watch how it functions. They can study exactly how it interacts with other cells, like the ones that line our blood vessels, and they can use that knowledge to create new lines of cells that produce incredibly powerful antibodies. It's a huge leap in understanding our body's own defense systems.
But as amazing as all this is, the technology isn't standing still. Not at all. The team behind it is constantly pushing the boundaries of what's even possible.
I mean, this timeline really tells a story of relentless innovation. It went from a semi-automated system back in 2013 to the fully automated and integrated nanopick we see today. The evolution has been so fast, and now they're already looking ahead to the next generation, coming in 2025.
And this next version, the NanoPick 2000, looks absolutely incredible. It's going to have a built-in incubator to keep the cells perfectly happy and comfortable during experiments, a completely sterile environment, laser autofocus for even sharper precision, and get this, an automated system for swapping out the tools it uses. They are literally automating every single step of the process.
So I guess I just want to leave you with this question. We've talked about cancer, the brain, immunology, but we're just scratching the surface here. When you have the power to isolate and listen to the individual story of any single cell you choose, what other secrets are we finally going to be able to uncover? The possibilities, they're just staggering to think about.